Hello, my name is Canru Han. I am a high school student at Beanstalk International Bilingual School in China. The paper I will be presenting today is titled Treatment for Heavy Metal Pollution, Remediating Soil Environment by Harnessing Genetic and Biochemical Capabilities of Bacteria. These are the contents that I'm going to present today. First, I'm going to be talking about the disposal of heavy metals. Heavy metals generally exist in complicated precipitated forms and insoluble conditions, such as those in mineral formations. Because of high absorption capacity of the soil, naturally existing heavy metals are not easily accessible to living organisms. Unfortunately, heavy metals are causing significant pollution because of human activity are now accessible to living organisms. The elements that can affect the accumulation of heavy metal ions in the food web include the sources of heavy metals, concentrations in the soil features, and the degree and the amount of uptake by plants, and the level of animal absorption. When heavy metal concentrations in the environment raise up to over permissible limits due to the biogeochemical cycle, all living things may be at risk. Traditional methods to deal with heavy metal pollution are all primarily physical or chemical, such as chemical precipitation, ion exchange, etc. Although they have some advantages, they require high costs, high energy consumption, and are easy to produce secondary pollution. Since the 1980s, bioremediation technology, especially bacterial remediation technology, as a new method to treat heavy metal pollution, has attracted widespread attention due to its low cost and high efficiency. I'm going to talk about redox transformation of heavy metals by bacteria now. Bacteria can alter the valency state of metals to change and reduce their toxicity, solubility, mobility, and other properties. For example, acidophilus can convert highly toxic chromium 6 plus to chromium 3 plus, which is a less toxic and soluble state, through its metabolic activities, thereby reducing the hazards of the chromium ions. Some other bacteria can reduce the insoluble plutonium 4 plus to plutonium 3 plus and reduce mercury 2 plus to its elementary substance of mercury, which reduces the toxicity. Now I'm going to explain the absorption and adhesion process of heavy metals by bacteria. The fixation of heavy metals by bacteria mainly relies on three standard methods: extracellular compactation, extracellular precipitation, and intracellular accumulation. Bacteria's cell wall contains functional groups like sulfur-hydryl, phosphorhydryl, hydroxyl, and carboxyl groups, and these groups contain atoms such as phosphorus, sulfur, and oxygen. These atoms have lone pairs of electrons that could provide the coordinated complexation for heavy metals, allowing heavy metals to be absorbed by bacteria's cell wall. The absorption strength of bacteria to heavy metals is affected by temperature, pH, nutrition, etc. Therefore, more investigations should be carried out to maximize the absorption capacity of bacteria and further explore their absorption principle. Now I'm going to talk about the relationship between mycorrhiza and bacterial remediation. Mycorrhiza is a symbiotic phenomenon. It is the combination of soil fungi and higher plant roots within a mutualistic relationship where both organisms benefit. The mutualistic interaction between mycorrhizal fungi and plant roots can be regarded in three ways. Firstly, because heavy metal hyperaccumulation plants can absorb and transfer heavy metals, the toxicity of heavy metals to soil bacteria is reduced. Moreover, the plant root system can not only provide compensation with heavy metals through the secretion of organic acids and amino acids, but also can provide nutrients for mycorrhizal fungi living so they can improve their activity. Secondly, the metabolic activities of the mycorrhizal fungi secretes organic acids and extracellular precipitation, extracellular complexation, and redox activity of heavy metals are happen because of it. As a result, the toxicity of heavy metals is reduced, and the toxic effect of heavy metals on plant roots is also reduced. Finally, the existence of mycorrhizal fungi can significantly improve the activity and growth of higher plants. Especially in soils with poor nutritional conditions, mycorrhizal fungi can provide the plant roots with resources necessary for their growth, like water, nitrogen, phosphorus, zinc, through the vast Hyphae network. Currently, the main difficulty in mycorrhizal restoration is obtaining bacteria with strong restoration abilities and suitable plants according to the relationship. Now I'm going to present a case study. The case study is named Bacterial Remediation of Heavy Metal Polluted Soil and Effluent from Paper Mill Industry. This is an experiment done to investigate whether proteobacteria or non-proteobacteria are better at resolving heavy metal pollution. Regardless to their division of phylum, they're all bacteria and all have the genetic and biochemical capabilities to resolve the issue of heavy metal pollution to a certain extent, and are all essential implements under molecular biology. The contaminated and uncontaminated soil and water samples were utilized for the bioremediation study. The experiment consists of 3 trials. Treatment 1 treated with proteobacteria, treatment 2 treated with non-proteobacteria, and treatment 3 without any bacteria, which serves as a control experiment. The study has shown that some bacteria have the tendency to survive and transform heavy metals to be less toxic. Though the soil effluent discharge was not pre-treated, the study showed that bacteria isolated from the paper mill were capable of absorbing highly toxic heavy metal accumulation in a time-dependent manner. This is possibly the most suitable waste management process in paper mills. The application of this method is a continuous system, so bacterial bioaccumulation of heavy metals and molecular characterization are among the future plans of research. Finally, I'm going to end the presentation with some prospects. The prospects of heavy metal elements in soil, such as mobility, valency states, and toxicity, are affected by soil properties. Bacteria are essential components in the soil environment, and their metabolic activities directly impact the soil's physical and chemical properties. Furthermore, bacteria themselves can also act on heavy metals through a series of activities. Furthermore, bacteria themselves, these mechanisms that bacteria carry out can be a radically based bacterial remediation of soil heavy metal pollution. Researchers have extensively studied the mechanism of interaction between bacteria and heavy metal ions, but there are only a few researchers on the interaction between bacteria and rare earth elements. Due to years of mining in irregular earth mining areas, severe ecological problems with heavy metal pollution have been raised on varying degrees, posing threat to farming, animal agriculture, and domestic water in the mining areas, and are causing a very huge environmental risk. Through the effect of bacteria on heavy metals, we discovered that it is a feasible solution to control heavy metal pollution. Therefore, in future researches, we should focus on the effect of bacteria on rare earth elements to find a possible way to control pollution in the occasional earth mining areas. Besides, if bacteria can be manipulated through gene engineering to increase their ability to remediate heavy metal pollution, the treatment process on this issue can be carried out with higher efficiency, thus resolving it in a higher degree. That's all. Thank you for listening and watching.